this series, we speak with scientists from the Dangoor Center for Personalized Medicine to gain an up-close understanding of what happens in their research laboratories, which developments are expected to reach the market, and how medicine will look in the coming years. This time, we're hosting Professor Erez Levanon. He's from the Goodman Faculty of Life Sciences at Bar-Ilan University and a member of the Dangoor Center for Personalized Medicine. Professor Levanon's field of research is computational biology. His laboratory focuses on genome research with a particular emphasis on a phenomenon known as RNA editing. Professor Levanon completed his master's degree in the interdisciplinary program at Tel Aviv University, where he also wrote his doctoral thesis. He followed this with a postdoctoral fellowship at Harvard University and returned to Israel in 2009 to join Bar-Ilan University. His research has earned him several accolades, including the Krill Prize from the Wolf Foundation and the very prestigious ERC grant from the European Union. He currently leads a genome research and computational biology lab at the Faculty of Life Sciences at Bar-Ilan University. Professor Levanon, welcome. Thank you for joining us.
Very happy to be here.
So let's start with some of the basics. What is RNA editing? Your lab focuses on this. What is RNA editing, DNA? Give us a little bit of background.
Okay. Do you need some background about what is DNA and what is RNA?
Sure, let's start with that.
So if you think about, let's think you are a cook and you want to cook something in your kitchen. And you have a book with all the recipes in your library, and you want to see to choose one of the recipe to prepare a meal based on the information that you have and the instructions you have in the book. But the book is very costly, a lot of money, and very delicate. So you copy one of the recipes that you want to cook into a page of paper, and then took it to the kitchen and start following the instruction and doing this specific dish. So the book is a DNA. You have all the instruction how to encode something, and I will elaborate more in a few minutes. And the page is the RNA, is a copy of portion of the book with specific instruction to do something. So if you think about our body is comprised from 10 in the power of 13 different cells. A huge number of cells with huge many functions that each of them is doing all the time. But the basic is that in every one of them you have a software, a code of instruction to build proteins and to build other component in the cells that doing their work. So the DNA is, as I mentioned, is the book with all the recipe, and the RNA is the instructions that currently the cell is now preparing. Based on the instructions that you have in this paper on the RNA, you can manage and prepare your meal. So this is different between DNA and RNA.
So the DNA produces different kinds of RNA depending on the needs of the cell or the body?
Exactly. So the DNA is exactly the same in all the cells in our body. We have about 3,000 different genes in the genome. The genome is about 300 bases per long, and portion of it are simply instruction how to produce a gene. And RNA is the instruction for produce specific protein from a specific gene. And the RNA editing is a phenomenon which is fantastic, and this is what I think it's fantastic because this is what I'm doing for more than 20 years now, is a phenomenon when the RNA, after it got copied from the DNA, can be altered and can be edited. So the content of the RNA can be changed from what the original source was encoded in the genome. So in one gene in the genome, you can potentially have multiple versions at the RNA level. So you can have one recipe in the book, of the cooking book, but many versions of it that derive, and so you can have a different version of the meals that you want to do. And this is basically the RNA editing in metaphor.
What kind of versions, like what kind of editing would you want to do?
Okay, so our basic content of the DNA is as I mentioned, is information how to encode a gene. And unlike computers that you have 0 and 1 as the language of the information, in biology we have 4 letters, A, C, T, and G. So all the genome is comprised from four molecules that we mark them as A, C, T, and G.
Does it stand for something?
Yes, for the specific nucleotide types that each of them is represented.
Okay.
So there's 4 alphabet genome. And the RNA editing is a protein. It's derived, it's created by a protein that you called ADAR that can change A into G. Actually, it's doing something that's called deamination that alters the adenosine, the A, into inosine, the I, but the I is read as a G. So from now on, I will going to refer as changes from A to G. Exactly.
Okay.
So you have something, in the genome you have A, but in some copies of the RNA, you have in some position G instead. And because this is leading how the protein is going to be translated, you can have a combinatorial number of versions of a protein. So you can have in the genome, let's say in one gene, 5 A's that can be edited. You have 2 to the power of 5 different versions of proteins that emerge from it. And this is, we found, and many others, that is critical to have this phenomenon. Without editing, we simply cannot live.
So your body edits it on its own and creates, so let me see if I got this right. The RNA that's created can be composed of 4 different kinds of proteins, and each RNA that comes out from the DNA could have a different amount of each of these 4 kinds of proteins to accomplish different things?
Basically, yes, but to make it more complicated, I have to say that majority of this editing take place outside of the protein region. They have a completely different purpose. So I will go now for what is really why it's so important. For a cell, for every cell in every animal, the worst scenarios that you can imagine is to be attacked by viruses. Viruses is very dramatic and can take control. Simply, they put their code in the DNA and alter the cell to produce new viruses. And our very sophisticated immune system is very, I would say, slow in order to have very efficient against viruses. So it's very good to have very good, complicated immune system, but because it takes time till the T cells and the B cells known, activated, and going to act against the viruses, the cell itself needs something that's going to be very dramatic and very efficient against the viral attack. So there is in every cell in our body sensors that every time, in our many cells, in every cell in our body, there is a sensor that look and tells the cell itself if it's now under viral attack. One of the sensors called MDA5 protein. And this protein, once it recognizes double-strand RNA, which RNA can fold off in itself, and it said we are under viral attack because almost all viruses have long double-strand RNA.
Okay.
So once it recognizes long double-strand RNA, it activates the innate immune system that works inside the cell. The problem is that our genome is comprised from a huge number of dead viruses or virus-like elements. So our genome produced virus-like particles all the time. So it's very dangerous that we are going to be
attacking our own.
Exactly, all the time. So the main purpose of this ADAR, this enzyme, and this editing is once there is something that look like viruses that come from the DNA and RNA that now look for inside the nuclear, it's edited multiple times, and now we will not recognize as a virus. So it can go out to the cytoplasm, to the outer region of the cell, and will not activate the immune system. So this is really the critical function of editing. So, and this is important for our next stage of the conversation, because it means that every cell in our body, and actually in every animal, have extensive editing that undergo naturally. So this machine is working naturally in every cell all the time in millions of positions in the genome in order to inhibit the false activation of the immune system. So what we are doing is how to manipulate this system now to, let's say, create a correct mutation. This is the technology that emerged from
this. If the body is fighting against itself, or So you threw out a term ADAR. What is ADAR?
ADAR is adenosine deaminase protein that act on RNA. This is a protein that what is doing is simply to do this editing, and it's act in every cell in our body all the time. And what we do in the lab is trying to manipulate it and convince him to go for another places that we want him to work. So for example, if someone is born with a mutation, which can be corrected by this manipulation, for example, G to A mutation. You know that mutation is a case where you have an error in the genome, and this error led for a malfunction of protein, and if its protein is important, people have genetic disease. So the traditional path, and traditional is funny to say because all this technology of correcting DNA is something very...
correcting DNA or RNA?
Correcting DNA is traditional.
Okay.
But it's funny to say because it's 10 years old, but in biology it's already old news.
Old, it's traditional.
Yeah, so CRISPR, which in DNA editing based on this amazing technologies that from the last decade of CRISPR that we can allow us now to go for the DNA and change the content of the DNA, which we called it DNA editing. But this process, when you change the DNA with the CRISPR system, it's somehow, you have some disadvantages because this system is depend on a protein that derived from bacteria, or the CRISPR technology derived from the amazing discovery of that won the Nobel Prize just recently. As the idea is that you can target specific proteins that we call Cas, that specific position in your genome, and by that alter it. But the problem is that you need proteins that you have to, if you want to use it in correct mutation of someone with a genetic disease, you have to introduce this protein from the outside, which is very difficult and also very problematic from immune point of view. It's bacterial protein. And also, the another issue is that it's systems that derive from bacteria, so it's not very accurate. In many ways, it can make another error in other places, and eventually leading for very devastating situation. Many mutations in our body can lead for cancer. This is how cancer evolved. So CRISPR, which is now the traditional, I would say, gene editing technology.
Old-fashioned.
Yeah, it's amazing breakthrough, and there are clearly some cases that we can use it, but what we are doing in the lab is something that is, I believe, is more safe, and we are not the only one, I have to say. There are many other groups in the world now doing the same, and the idea is to utilize the endogenous ADAR, the protein ADAR that
your own body produces, exactly. Endogenous, it's internal.
Exactly, so it's already activating every cell in our body, and simply convince him to go for other places. So how you convince him? By understanding what are the specific structural RNA features that he like, you can introduce complementary sequence from the outside and convince other to go for another place.
Hang on, I'm going to just stop you for a minute because you introduced a whole bunch of concepts here. I want to make sure that I understand them. So one of the things you mentioned was that the DNA produces this RNA, and then there's this intervening thing called ADAR, which helps edit the RNA.
Exactly.
And that's where you're talking about the 4 different letter elements, and the ADAR goes in and can help edit those.
You can change
an A to a G
in specific positions. Think about every RNA is a long string of letters.
Got it. So that's what the ADAR is. And when you're saying a lot of traditionally, there was this amazing thing called CRISPR, which was almost like a scissors for the DNA to edit the DNA. But there were problems with it. First of all, you mentioned a few that there are side effects potentially. And the DNA, I know, the DNA is a permanent part of your body.
Exactly.
The RNA, as you said, you could copy down the recipe many times. And if there's a mess up with one of the copies, it will go away.
Exactly.
And it's not a permanent change, whereas if you edit the DNA and there are all kinds of problems, you're now in trouble because it's there forever.
Exactly.
Versus the RNA, which the potential side effects are much more minimal because it's not a permanent change.
Exactly.
So what you're trying to do in your lab is to get involved at this intervening moment when the RNA is getting its messaging or getting its editing with this ADAR molecule. Is it a molecule? Protein, this ADAR protein, and say, we know what we need to do we know what the RNA should look like. In some bodies, it's mutated. It's not doing what it doesn't look like it should, ideally, for the body. So we're going to try to use the endogenous ADAR that's produced. We're going to come in from the outside and help convince it or change it or whatever language, I'm sure there's a better scientific term, adjust it, edit it, so that it creates the RNA that we want.
Exactly.
Okay, so I got it. Now we can move on. I just want to make sure I understood it as a non-scientist to clarify it all. So how do you do this? You said you introduce exogenous things to this endogenous, meaning from the outside to this protein, this ADAR that's created by the body. What do you do? How do you...
ADAR, this protein ADAR is, don't edit every RNA in the cell. As I mentioned, his main duty is to provoke, to inhibit the false activation of the immune system. So he looks for regions that mimic viruses structure. So if you see something that reminds him a virus, is going to edit it. So if you have an RNA, as I mentioned, have 4 letters, A, C, T, and G, and they have a beautiful, actually, for someone that learned biology, it's not A, G, C, T, in the RNA, the T is a U, but it's the details. I'm not going much into all these details.
You'll keep it easier for those of us who are not experts.
Okay, so the idea is that the beauty issue is that A is always connected to a T and G into a C. So you can build a complementary sequence, the complementary, where with specific feature like key and a lock that will bind to the RNA with this A to G, A, T, and G, C connection, and will form the structure that the other like. And by that, other going to see it and going to edit it. So the idea is to understand exactly where are the positions that editing take place in the human body. So this is something that we are doing for many, many years now. And based on this and utilizing the machine learning and many other approaches to understand what are going to be the best sequence that will lead for the edit, for when that we introduce it to the cell, it will bind for specific RNA and recruit the endogenous ADAR to edit the specific position that we want.
Got it. And this is specifically, I think you mentioned this kind of peripherally along the way, this is specifically to stop the body from attacking things that look like viruses.
So this is what is doing, this is main purpose, but now we want to utilize it for completely different arena. If, for example, if you have a mutation, and let's think that someone have a mutation of rare genetic disease, we can look what is the RNA that this protein that has the instruction to produce this malfunction protein. We can now introduce an RNA from the outside that will bind for this region of the RNA of the malfunction protein, and now the other will go and fix this mutation. So you can fix the mutation of people with endogenous disease. The issue is that you can do it even more than that. You can enhance human body. For example, there is a very famous mutation that everyone have it, a good mutation. There are also good mutations, cannot have any cholesterol, high cholesterol. And this mutation was explored in several decades now. The mechanism is fully understood. And the lucky person, about 1% of the population that have this mutation, will never have high cholesterol level. But we can do it for everyone.
Oops,
we can do it for everyone. There are even a company in the United States that try to do DNA editing, change the content of this DNA. The main market obviously is people with high cholesterol level, but if you can think for the future, you can think that everyone can get it. But now we're going for the risk in the DNA editing.
Right. Because you can hit other genomes, you can hit other things with side effects we were talking about.
And maybe we don't fully understand the biology of this.
But if you do it with the RNA, you get multiple chances.
Exactly. Or something that we now just start to think about. Humankind want to conquer Mars in the future, but we don't fit to live in
right. We're not built for. We're not Martians.
Exactly.
We're Earthlings.
So NASA has a project to try to think which gene we have to change in order to have to fit to live for many years with low gravity, for example, or with no gravity. And the idea, which is completely ethical, very problematic, maybe to create baby astronauts that will fit in the 100 years after we understand everything, and to engineer them from beginning to fit for low. So it's a huge ethical issue that obviously it's not a plan. It's just
Right.
ideas.
Well, anytime you talk about genetic engineering, even so, you take it to the good, right? Well, genetically engineer so that humans don't get issues with cholesterol. We could genetically engineer other things, and who decides what is the ideal and what should be engineered and how much is it's a whole.
So many of the questions, ethical questions, are also relevant for RNA editing, obviously. But many of the questions that are fit specifically for genome engineering and DNA editing are now less relevant for RNA editing.
Because it's temporary.
Exactly.
You're not actually transforming the genetic material of the body.
Exactly. You don't transform into the next generation. So many other, and if there is better treatment or better understanding, you can simply...
Right, you could do the next round of RNA. I never thought about that. I knew that DNA editing was a problem because it's more permanent, and if there's side effects, or you hit the wrong genomes, or you somehow mess up with what you're editing. I never thought about the ethical content, but it's fascinating that it also addresses that issue. One more reason you chose the right field, RNA, not DNA, editing. Right? You could be pleased with your choice there.
Yeah, definitely.
It is clearly the future, and then, you know...
We think so, but obviously there are many other angles for the same, but there are definitely room for everything, but for RNA editing, I believe there is a lot of room that are very relevant for many future clinical applications.
But you're a computational biologist. We're talking about real biology. Where does the computation come in?
So, first of all, what my lab and myself and everything that I work, I have to mention Professor Eisenberg from Tel Aviv and Shai Ben-Ari of Bar Ilan. These are close collaborators. So what we did for the last more than almost 2 decades now is to first to understand where editing takes place endogenously. So what our main contribution to the field was to mapping where editing takes place. So it was, and this is completely computational. You have the genome sequences, and we took many RNA sequences. The idea was to find where you have in the genome A, while in the RNA you have G. It sounds a trivial task for computational biologists, but because the noise level, simply the error of the sequencing is way higher than the editing, so what we really did is to develop some tricks to how to distinguish between editing and background noise. So what we did is to map where editing takes place. And one fun discoveries that we made and led with Eli led this project with Josh Rosenthal that, for example, octopus in their brain, they don't have exactly brains. Their nervous system have a huge amount of editing that change the protein. In human, we have many editing in non-coding regions that mimic viruses. But in octopus and other cephalopods, we have a huge number of editing that change the protein. So you can have a protein with, let's say, 100 positions with that can be edited. So you have 2 to the power of 100 different versions, a huge astronomical number of different proteins. So you have a huge number of different proteins in their body. And
in octopus.
In octopus, in squid, and we know the octopus are much more smarter
That's
than
right.
the other
There was just a movie about it. Did you see the movie?
Yeah.
Right? It was fascinating.
Yeah. So they are much smarter than other mollusca. Their intelligence is resemblable for many vertebrate or even mammalian, which are considered much more smart. And we speculate, and completely speculate, that this huge diversity that they have only in the nervous system is that derived from RNA editing contribute for this
intelligence that they have.
Exactly.
That's amazing. So the RNA actually can impact the intelligence of a creature, or the amount of editing that's happening. It's almost a certain kind of innate intelligence on its own to keep doing this editing. Obviously RNA editing have a lot of function in their brain, also in human brain. It's something that people know for many years ago, that the main function of the other protein families, it's actually have 3 members in the human body, ADAR1, 2, and 3. So I mentioned what I mainly discussed was ADAR1, but ADAR2 and 3 work mainly in the brain. And in the brain, they change ion channel, proteins that are very important for the creation of action potential and how they actually to activate neurons. And we know that without proper editing, the brain simply not going to work properly. And this is something that we also work and study, and what exactly is the location, what is the importance of the editing in the brain. So evolution not only take the ADAR as a machine to avoid false activation in the immune system. Once you have these machines that can
alter the content, it also utilize it to diversify our neuronal activity.
Fascinating. So the computation piece, it's not just that you could go through many larger data sets. It's also to hold things constant. You can sort of, as you say, get rid of the background noise.
So the idea is that there is a huge number of, sequencing now is became very cheap.
Yeah.
When the human genome project was sequenced, it was billions of dollars to sequence genome of one person. Now it's less than $1,000. So million-fold less, which is remarkable. And so there is a lot of data of people that sequence DNA, also sequence RNA. So we collect RNA data that would sequence across all the world and looking for positions that we have chance for editing. And for example, one of the discoveries that we are very proud about is to see that in cancer, you see alteration, elevation of editing in almost all cancer type. So this means, if you think about it, that if you have high level of editing, it means that the immune system is more relaxed, because this is the main function of ADAR. And once we find that, once we understand that this editing is elevated in most of the cancer, we were thinking that if you reduce the editing, we can enhance the activity of checkpoint inhibitors. We know that there is now immune therapy is a very big deal in cancer, but it works for only small portion of the people and also for several type of cancer, not for all of them. So it's huge progress, but currently working for a limited number of percent. So what we show together with several labs in Boston, that when you reduce the editing, you increase the activity of these checkpoint inhibitors, of the immune therapy. So now there are several drug companies that working of how to inhibit the other activity in order to activate and enhance the activity of the immune therapy.
What's the connection? Why does lowering the number of...
Because immune therapy is, and this is really major breakthrough in cancer, immune therapy is to activate the immune system against the cancer.
Okay.
Immune therapy, our immune system is fascinating. It's work very efficient and actually work in our lifetime against most of the cancers we are developing. But obviously, people die from cancer. People get sick. And the major breakthrough in the last few years, another Nobel Prize, was that you can recruit and enhance the activity of the immune system to target and kill cancer.
The natural killer cells, right?
Not exactly, but in a way. But
Okay.
it's a natural. It's a natural. Literally, it's natural killer cells in a way. The T cell. So the idea is to understand how the cancer is try to avoid the immune system and or to eliminate it. This is the PD-L1 and PD-1 proteins, or to activate and to train the immune system specifically against the cancer. But here we have some mechanism that we know that editing is main duty is to reduce the immune system, because it tells the cell, don't worry, you are not under viral attack. So the virus, if the cancer want to have this in high level, because this relaxes the immune system. So if you remove the editing, the immune system is much more aggressive.
Right.
And this is the idea. On the other end, if you have not enough editing, it means that now your immune system is overactivated. And this is paper that we just published, led by the group of Yuval Dor from the University, that if you take, I will take one step back. We are thinking now about a completely different arena. This is the fascinating.
But it's all connected to what you're doing.
So we know that type 1 diabetes is a disease very common, but we don't understand what causes type 1 diabetes. Why some people have type 1 diabetes and others don't have it. And people know for many years that the initial step of type 1 diabetes is something that looks like that there is antiviral response in the beta cell. But people spend their career to find these viruses, and they didn't find any virus in the pancreas or in the beta cells of people with type 1 diabetes. And what we suggest that maybe if you have alteration in your editing, so you don't have enough editing, now the cell sink is under viral attack. And it starts to attack these cells. So Yuval, in his group, what they did, they create mice without editing activity, without ADAR in their beta cells. And this mice developed type 1 diabetes. And this was remarkable because this is really a breakthrough in understanding why autoimmune disease can occur. Simply if you don't have enough editing, you have load of virus-like element that produce from your own genome.
And your body fights against it.
Exactly.
And it's applicable, it seems like you have to find the balance
Everything
here
is balanced,
with addressing every disease. But in diabetes, it seems like there's not enough, and whereas in cancer, there's too much.
Exactly.
So it's really, and that's where I guess, you're a member of the Dangoor Center for Personalized Medicine. That's really where the personalization comes in, right?
Yeah, but also for people with specific mutation. Now you can now going back for the engineering process, now you can design specific guide that will fix specific mutations that specific person have. There are about 6,000 different genetic diseases, but each of the diseases have large number of different mutations. So you fix one mutation, it's fit for only a small group of this population. So the idea is to, as you mentioned, do personalized medicine for each of the mutation, or globally, and also in different cancer, or also in the autoimmunity system.
Does your lab focus on all of this, or so just the basics, or? We
focus on RNA editing, and this journey was started as completely fun project that we know that we have the tool to solve it, simply to know where editing takes place in the human genome. And it's so exciting to see how it took us from cancer to one side, autoimmunity, to now engineering of and guides that can change the content of our genetic information, and also for octopus or in coral. And so
it's amazing,
yes,
the connection and the growth. And I'm sure for you it's really exciting to see some of the more basic research you're doing applied to really change people's lives, to address these kinds of mutations in their body.
Exactly. Now there are several companies that work on this. So it's really very vivid place and in science, and very exciting period on this specific topic with so many opportunities, which is really exciting.
So back up though, how did you get involved in this particularly? What made you choose RNA editing to begin with, before we were where we are now? Why did you find it exciting then?
It's simply because I was an undergraduate student many years ago, and in parallel I work in a biotech company that's called Computational. It still exists, but it's very different. Yes, it's very different for now. Back then it was computational biology with a very massive and very powerful platform to do genetic research. And this was before the genome was even published. And we have the best platform to analyze RNA. And this was one of the main products of the company. And I simply heard a lesson by Shula Michaeli, you may know her.
I know Shula very well.
When
Former Vice President for Research.
Exactly, when I was an undergraduate student. And she mentioned this phenomenon that the RNA editing, that there is something, and people know about 2 places in the genome that it takes place. And I realized that with our platform in the company, we can find where it's actually take place. And together with many great friends and utilizing platforms that already was there, we managed to find where editing take place. And I was thinking that is simply a project of, I was undergraduate back then. So it's going to be a project, and I simply
You just kept going, just kept going. And Shula can take credit for inspiring you.
Definitely.
Do you work with Shula these days at Bar Ilan? I know there's a good group of people working on RNA.
We speak a lot. Whereas now we're thinking about projects to do together, but we are definitely.
Right. Well, there are a number of professors at Bar Ilan who work on RNA. stuff, which is amazing. That Bar-Ilan really sort of figured out, and all of you, obviously, as professors there, this is the field of the future to a large degree.
Yes.
You'll accept it. Yes, of course. I'm definitely at the forefront of the future, but you are with what you're doing. And
everyone in the science is in the forefront, but we have the luck that now it's also became important. So everyone doing, every lab, every PI is exciting about his work, but we just simply have the luck that is became also the path how to utilize what we do to even to the clinic is now became not a science fiction, but something that we see already results.
It does seem science fiction-y, though. It really does. And the RNA protein seems like it was almost designed because of its properties, in the sense of that there is this intervening protein, the ADAR, that you could get involved with. It's temporary, the RNA. It can be produced over and over. Almost you could test different things without major consequences. It's such a great response to the problematic, CRISPR editing of the DNA. It really seems designed for...
It's designed, obviously. This is exactly, this is how life is working.
That's right, our bodies have designed it.
Exactly. So in order to survive, you need flexible machine, and RNA is the best place to be flexible and change it. And obviously, it's not only in human. RNA is the best arena to play with and to have a lot of combinatorial that can different cell. You have the same genome from in all the cells in our body. But every cell is very different. Cell from the brain is very different from cell from the lung, or cell in the brain during specific condition is different than cell in the brain in other condition. So RNA is obviously the place you want to play and to do a variation. The DNA is fixed, so RNA is a playground for most of the modifications.
Right, and for scientists to be able to explore without worrying about what you're doing permanently to DNA. So that's amazing. Where do you see your lab in 5 years, 10 years? What's your dream? Where would you like to go with it?
We hope, now we're doing with several people. Gal Oren is the leader of this field from now is Stanford, and to understand, to have the ability, once we know which are the mutations that we want to fix, what is going to be the guide that's going to send. This is a very difficult task, and I really hope that, and we play a lot of effort on this, and I really hope that we're going to solve it, because this means that the path now for a clinic is much shorter. So because now can take a year to design this guide, but if you can do it completely artificial by computer, it's something that I believe will change all the industry of drug development, because you can simply find the edits that you want to do, and immediately you have the proper drug. Also, I really hope that we understand more and more about the endogenous editing activity. What is important for? We build an atlas of editing the change coding sequence in more than 1,000 places. So to understand what are the functions, to understand the editing connection for autoimmune disease is something that we work very hard about, which I mentioned, type 1 diabetes, but we now work in additional autoimmune, because autoimmune diseases are a complete enigma. And we think that we find something to push, to thread to push, and to maybe it's going to be solve this enigma.
Autoimmune diseases are when the body's sort of fighting against itself, right?
Exactly.
And so if you can stop it, it sounds like you understand the process behind it to some degree, begadol, in big picture.
Yeah, but obviously there are many other elements, obviously.
Right.
But this element was underseen. People didn't recognize the importance of it. And this, when you have new player in the game, you can look at it in better perspective and understand it much better, because currently the success in fighting against autoimmune disease is relatively modest. But if, and I hope that this element of editing is really crucial, in the basic, it gives us a lot of hope that we will be able to develop better drug in the future.
And your background in computation, actually, as you said, allows you to almost experiment on a computer, or like to look at all the data, to try different things, to map it out without having to have a human body that you're testing it on, at least in the beginning.
Yeah, so at least some of the aspect is completely computational. To find editing is completely computational. To design the guide RNA is completely computational, but obviously everything that you find computational, you have to do the experiment.
Of course, but you are able to, as you said, by running these computers and I mean, I don't know enough about it to even use the right language, but to run these tests, to look through the data, to figure it all out, can be done so much, not just quicker, but more safely than trying this on a person and trying that on a person and trying, you at least know where to begin in a shorter period of time, as you said.
Definitely.
And so it's amazing that you managed to take two different, very, very different fields, biology and I mean, you obviously are brilliant in both computer, stuff and biology. You need both of them together to do this.
It's actually now a very common theme in most universities in the world now have a field of computational biology.
Right.
Bar-Ilan was, I believe, based on the visionary of Ron Unger, was the first in Israel to have a program in computational biology. So we have, I believe, the largest program in Israel of computational biology. There are 20 or 30 students every year that studying both biology and computer science and fit to enter to this game.
To use the AI, the technology that's out there to actually push the field of biology forward tremendously in ways that you can't when you're contained by a physical body or physical molecules or cells or whatever it is, which is really amazing.
Yes, biological become a science of big data. There's so much data and
well, if you have the data out there, at least you know what to do with it, which is, it's a wonderful thing. And it, I guess you take from the big to the small, meaning you could look at all the big data and then address even orphan diseases, which are, the N is so small that in some ways to invest in finding a solution for just that is a challenge financially, but if you could look at do a whole bunch of the work on a computer, then you can address the big numbers of autoimmune diseases like type 1 diabetes and some of these orphan diseases that are also autoimmune in the same way, right? Do you have a moment in your lab where you went, oh, it's all worth it. This is what I'm doing. I know I'm in the right place at the right time.
No, I feel I don't, but I think all the time.
Always, I love it. You wake up every morning and you're excited. At the end of the day, you think.
There are many other challenges in life, but yes,
but in your lab, it's always exciting.
Yeah, definitely.
And you have wonderful students that you work with.
Yes, they are great students, and actually, I have to be fair. I don't do nothing. So all the research the students are now doing, I just try to
guide.
Yes, or not to interfere. Yes.
Oh, I'm sure you do more than just not interfere.
Yeah, but they are doing all the work.
Well, look, it's many people that you're guiding and directing and helping to actually do a lot of it.
But they are doing the work, so they have much more fun than me because I have to do all the bureaucracy, but
you have to apply for ERC grants.
Yeah, but yes.
Right. That's on you. So both are necessary, right? And by you guiding the students, we're gonna see amazing results out of your lab. I am sure. It's an exciting moment to be in the field of RNA editing and to see the potential across the board. And you get to, it sounds like, be involved in some of the basic research and then see it mapped onto transformational change in people's lives. That's
Hopefully,
wonderful.
yes.
Hopefully. Well, that's the goal, right? So amazing. You should continue guiding your students properly. If you're not going to take the credit, I'll give you a little bit more of it. And hopefully well, it sounds like you have shared projects with so many labs, and that's also you building those connections across the world and working together with people who know about oceanic creatures and computers and biology and all of this together. So it's amazing. Thank you.
Thank you.
Thank you for the work you do, and thank you for being with us here today. This was really fascinating and really moving us forward in the world of science. So appreciate it.
Thank you
very much.